Hello, hello, hello. Okay, it should be working now. Okay, yeah, it's picking it up. Okay, awesome. All right, sorry. While you're here, this projector, let me make sure I can get the projector to work because, okay, it looks like it's gonna work. Okay, never mind. We're good.
It's not up to you.
Okay, cool, cool. Thank you. You have to disable the privacy function on it, so it's not something I can do, just to be clear. Only IT has access to that stuff, so it's a good time. Huh? I don't know. I'm just assuming that literally nobody else actually runs their stuff through Zoom or something like that while they're offering their class, which I'm, whatever, that's their choice, I guess. But I do it literally so that if you guys wanted to Zoom in instead of come in, and I guess other people don't do that stuff. It's what, right? And it's different. Maybe it's just different. My settings just were set differently on my profile or something. I don't know, but it doesn't really matter for you guys. Okay, so we're gonna pull up the Kahoots. Finally. We're only 1 minute behind, so we're doing good. Okay, cool. It's so bright behind me. It's been doing this at every computer that I've used so far this week. Telling me it's an unrecognized device, even though I used it last week. All right, we got chapter 2 first, and then we'll do chapter 4 after that. Chapter 2 first, then we'll do chapter 2 activity. Then we'll do, yes. All righty. There we go. All right, awesome. I know some of you still getting saying we got 10 of us, I think, today. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? Yeah. So there's 14 people enrolled in the in the section. Let's see if there's anybody online. Nope. Just me. All right, so that should be all of us that are here in the room. So we're going to go ahead and go through this. So that you guys know, first of all, if I made a comment on your concept map from saying, hey, you forgot to add so a lot of people forgot, at least like maybe one, a lot of people may have forgotten on one concept map to add their reflection question. That's a pretty common thing because I know we're trying to just knock it out and whatever. Forgot to add your reflection question, add it today. Just add it today at some point by midnight or something. Add it in the comments section or add it into re-upload your thing. Either way, And then I will go in and check all of those tomorrow and update those scores. So if you did them, I'd hate for you guys not to get full credit for the thing, right? Because the big thing is the concept map itself. But what you'll see more and more, especially in this class, it's not enough just to have the info. We have to be able to apply it. And so that's what the whole point of those reflection questions is to get to that point of like relating why we're learning this, right? So if you haven't done them, get them. If you haven't submitted your concept maps, which may or may not apply to somebody, then if that's you, or if you just submit 2 of them, but you want to knock out the other ones, you can still submit the other ones for partial credit. I'll only count them a day late. That's better than no credit, right? So if you haven't done them, get them done. All right. Even if you get them to me tomorrow, I'll only count them as a day late. I know it was a lot to get done. Okay. So we're being a little lenient here. Realize this week, we don't have any concept maps due. So I know the first week was a lot, but most weeks won't be like that. We got over that hump. Most weeks, so this week we don't have any, but most other weeks we'll have 2, maybe 3. And when I say 3, the 3rd chapter is much smaller. So I'm talking about chapter 25. So 9 and 25 I combined together, but 25 is super short. And so that should be a tiny concept map. And that's in unit 2. That's not even in unit 1. So we got like a while for that. Okay. Most of the time it'll be that. Rarely, like the first 2 labs, lab 0, 1, and 2, have some extra online lab stuff. But like lab 3, there's 1 online thing. Lab 4, 1 online thing, etc. So most will only have 1 after that until we get to the second 8 weeks. They come in a big lump. Okay, so second 8 weeks of the class, we'll get 5 at once, and then have none for a couple weeks, and then have another 5 all at once, and then none for the rest of the time. Okay. And they're like the other ones. It shouldn't take super long. The online labs, they don't take that long. Once you figure out how they work, they're pretty easy to get through. So, but anyway, so what I'm saying is that shouldn't be super taxed, not like the first week. Every week is not like that. Okay. All right, so we're going to see why we made our concept maps before coming in because this is our first day doing the thing, right? So here's our first question. What type of chemical bonds involve sharing electrons? All right, so our first question, what type of chemical bonds involve sharing electrons? The answer was covalent bonds, okay? Is Brooklyn here today? Brooklyn Tillman? Nope, not yet. Anyways, if I call you and you're not here, I come back to the names once I go through everybody, okay? So you get multiple tries. So if you do come in a little late, there is still time to get called on, all right? So next one is, I'm gonna try Asley? Asley, okay, Asley. Okay, it sounds like it looks good. That's kind of comforting. Asley, I'm gonna ask you, we know covalent bonds share the electrons. In ionic bonds, what happens? So giving up, donating, or transferring. Yes. Okay, right. So we're completely giving those. So my example in the lecture, if any of you guys actually watched the lecture, and I'm not judging, like I said, I know it was a lot. Okay. What we learned about was actually draw it on this screen. Make it big. Find our pen. Here we go. All righty. So where is it? Here it is. Okay. So with our chemical bonds, if we draw something like this, O linked to H's, this solid line here is our covalent bond. That means we're sharing electrons here. Now, if we have water like this, is the sharing equal between the oxygen and the hydrogen? So this is the big thing, okay? So oxygen is what we call electronegative. What this means is negative. What kind of charge do electrons have? Negative. Okay, so we're at least there, right? It hogs. What this means when it's electronegative is it hogs our negatively charged electrons. So it's going to spend more time with the electrons. In my example that I give in the video, I talk about it like sharing Barbies. Right? So I'm the big sister and my little sister Erica we might have to share Barbies. Now, as a big sister, do you think I was fair with that Barbie? You think I'm going to play with the Barbie more if I want to? The same with oxygen. Oxygen's hogging the Barbie. Okay, spends more time with the Barbie than the hydrogen does. So that leaves that oxygen having a negative aspect to it. We call that aspect polarity. If we're being real chemical, we'd say a dipole. Okay, in this case, we're just calling it polar. It's got a negative polarity. So that if it's negative and the hydrogen doesn't see the Barbie as much, which is our electron, which is negatively charged, if it doesn't see that enough, then what kind of polarity does it have? Positive, right? That's true for both of our hydrogens because oxygen is that greedy. It really does hog those electrons that much more. So we've got positive polarity on our hydrogens and a negative polarity on our oxygen. So it's unequal sharing of that. Sometimes, and you might have learned this in chemistry, sometimes that oxygen gets so greedy that it'll completely steal an electron. So you'll be left with an H plus and an OH negative, right? The O negative being on that oxygen. Because it stole an electron completely from one of the hydrogens. So we know water can kind of cycle between those two. Now, this is going to be imperative when we talk about pH. If you don't know why this is imperative for pH, don't worry. I'm going to show you because I know it's been a while, okay? So keep this in mind. This is what water does. It alternates between H2O, which is what we had going on over here, right? It alternates between H2O and H plus with OH negative. If you're water and you're pure water, whenever you split up your H2O into an H plus and an OH negative, you have equal amounts, equal numbers of H pluses and OH negatives. We can agree on that. If you're only water and water is the only thing in there, if you split one water molecule, you get one H plus and one OH negative, equal numbers, right? That is why on a pH scale of 0 to 14, that's why 7 is the middle where water is, and that's neutral. Because you have equal numbers of H pluses and OH negatives. That's why. It falls right in the middle. When you go towards 0, you actually have more H pluses. When you go to 14, you actually have less H pluses. Let me show you why it only goes to 0. Okay. I'm never going to make you do the math, but we have to look at the equation to understand what's happening. Okay, so bear with me. pH equals the negative log of the concentration of whatever your hydrogen ions are, whatever that concentration is. Okay, so we all know the back brackets means concentration, right? Okay. Know that. What does log mean? Does anybody know? Just generally. Logarithm. What does anybody know what that means? It means based in 10. That's it. That is literally it. Do not read past it, past that. Okay? That really is what that means. It's based in 10. Anybody know what the negative means then in that context? Inverse. So we're getting somewhere already. Inverse. So when we think of 10 to the negative 2, what's really the same thing as 10 to the negative 2 is the same thing as 1 over 10 to the 2. It's the same thing. Those are the same. So when we say a pH equals 2, what we're really saying, the negative log of the concentration of the hydrogen ions is 2. That's what we're saying. So what that really means is that we have 1 over 10 to the 2. Same thing as 10 to the negative 2. So if I look at a pH of 2, what's another way we could write that? 1 over 100, right? All right. What is pH of 0 then? If the pH is 0, that would be 1 over 10 to the what? 0, right? Anybody know what that is? Yeah, it's 1. You can't get past 1. That's it. There's no going past that. What that means is when the 1 is the answer to that, that's the concentration. What we're saying is 100% of your solution. If your pH is 0, that means 100% of your solution is entirely 100% hydrogen ions. There's nothing else in it. That means 100% of your solution is hydrogen ions. That's what a pH of 0 means. So that's why we can't get less than that. Because you can't get more hydrogen ions than 100% of it. It's impossible. That's why the end is at 0. All right. So coming back to the basic part of it. Get rid of this real quick. I didn't mean to get rid of that part, but it's fine. It's gone now, I guess. 10 to the 2. This is the exponent. Okay. So if pH and 1 over 100, can we agree that 1 over 100 looks like this? Okay. So we can get there. Okay. I know my math is terrible too, so I'm not judging if your math is bad. Okay. So if you have a pH, we're going to try. Okay. pH of 10 means 1 over 10 to the 10, which means 0.0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? There's another 0 and then 1. Yes. Which is bigger? 0.01 is bigger, right? This is your hydrogen concentration. So now you can see why lower pH means more hydrogen ions. It's because of that negative log BS. It's all in relation to the tens. So 10 to the negative 10 is pH 10. 10 to the negative 2 is pH 2. Same thing we say 10 to the 7, sorry, pH 7 is 10 to the negative 7. That's where we have equal amounts. Okay. So that is where neutral is. Okay. Are we tracking a little bit? Like I said, I'm not going to make you do the math, but keep the perception of what's going on. Because while I might ask you, and y'all are so good at recalling that less than 7 is acid and greater than 7 is base. They'll get you one question on my test, maybe. But what questions I might have on my test might be something like, your patient comes into your hospital, their blood pH is 7.0. And I'll tell you too, just so we know, I'll tell you too that the normal body pH is supposed to be between 7.35 and 7.45. And then the question would be something like, do they have more or less hydrogen ions in their blood than they're supposed to? More. Exactly. You guys tracking where we're getting this from? Okay. And so now we can put this into perspective of the numbers. Each pH means a factor of 10 difference in the hydrogen concentration. Okay. All right. So I'm going to leave that there because we're going to get to pH stuff here in just a second. So we had Aisley or Asley? Asley. I'm going to mess it up. I'm sorry. Okay. All right. So we got good on that. Okay. Let's go to our next question. I'm going to leave this up. Yes, you can cheat a little bit with it. Okay. If you can still read it on the next slides. Okay. All right. Next question. It might be about pH. Hydrogen bonds. Ooh, even it's related to pH. If y'all don't see how it's related. Hydrogen bonds. Tell me about them. Which of these apply? Be careful and actually read the answers. People get this one wrong. Read and understand the answers. Okay. So oftentimes we're getting the yellow chosen on this one. We'll go over why that's wrong in just a moment. Okay, do we have another one that it's so difficult for me to pronounce? Is it CeeLo? CeeLo, is she here? I don't know how to pronounce the name. I might be saying it incorrectly. Doesn't look like it. Okay, Tonya. You're okay. You're okay. As long as you get one question in, I count you guys as participating on this, okay? But if you want to re-enter, the code will be at the bottom. If you ever can't see it, just holler. It is 650387. Okay, so I'm going to pick on you, though, by the way. A hydrogen bond. Okay. So when we have a hydrogen bond, is it between two hydrogens or is it between a hydrogen and something else? Okay, so when we think of this, one of the most common ways to look at a hydrogen bond, we'll put it in its own little area over here, okay? And it's related because we're going to talk about it in relation to all of this stuff is related to each other, believe it or not. We hate it, but it is, okay? If I drew water up there, which we drew up there, but I'm going to redraw it. We know that H's have a positive polarity and the O has a negative polarity, right? And that in water, the whole thing that creates surface tension in water, and why we get beads of water on things, or the way that it balls up whenever rain's falling out of the sky or whatever, is from that surface tension, right? And that surface tension is created from this. Your H's from another water molecule attract to the O's of a different molecule, right? This here is a hydrogen bond. It's between a hydrogen that is having its electrons hogged by another atom, and it's almost always oxygen. Okay? Almost always oxygen. In biology, it's pretty much always oxygen. Okay? So that hydrogen's not getting its electron very much. It has a positive polarity, and it's attracted to something that has a negative polarity. Usually, oxygen that's hogging another hydrogen's atom, electron. Okay? We're tracking. So if we say it's formed between hydrogen atoms in different molecules, we got to read and understand that statement. It's saying that we have a bond between hydrogen atoms of different molecules. Is that what a hydrogen bond is? So this is a thing where the semantics of this stuff tell me that this doesn't sound like the crap that they pull in the nursing program. Because they'll put questions that part of it sounds right, but you have to put it into perspective. Or it might be right for certain situations, but you have to know for this situation what is right, right? So here our answer is holds water molecules to one another. Now you guys were probably, when you're answering this, trying to look for the definition of a hydrogen bond. We got to get away from that too. Because here, when we're looking for a definition type thing, we're going to fall into the trap of the yellow guy. And we can't allow that because the yellow guy wasn't actually correct. The semantics were throwing us off with it, right? When we see form between hydrogen atoms in different molecules, we think between a hydrogen and a different molecule, but that's not actually what the sentence says, right? It says between hydrogen atoms in different molecules. So we're talking about two hydrogens. Now, I try to make my test questions a little clearer than this, if I'm being honest with you, okay? But at the same time, we have to be able to break down and understand all of our choices, right? So that's why that one was wrong. And that's why hold water molecules to one another is right. Now, that may not be the definition of a hydrogen bond. And do hydrogen bonds, do they always have to involve water? No, absolutely not. In fact, tell me. Oh, I forget this doesn't automatically do the thing. Tell me what this looks like. You already know what this is going to be. DNA. Okay, we got that. So the DNA, we call this the DNA double helix, right? It's DNA. If we break down DNA, we unwind that ladder. We've got A's and T's, and we've got C's binding with G's, right? And that's what that ladder really looks like. What's this represent? Hydrogen bonds. In fact, your DNA, both of your chromosomes, both of your chromosome pairs for all of your pairs, so 23 pairs, right? You have 23 pairs of linear chromosomes. Those chromosomes are double-stranded DNA, wound up like that, right? All held together by the A's and T's and C's and G's by hydrogen bonds. And hydrogen bonds are just attractive forces. That's it. We're not exchanging electrons. We're not moving electrons, nothing like that. Where the hydrogen bond is just an attractive force between a positive polar hydrogen and a negative polar, usually oxygen, almost exclusively oxygen. All right. So when we start talking about adding acid to something, if I add a whole bunch of H pluses, we add acid, which is high in H pluses, right? Excess of H pluses. You see how these H pluses can interfere with stuff like this? So if I pour sulfuric acid on you, what would happen? Your skin would literally just burn off, right? But what's actually happening at the molecular level? We're breaking hydrogen bonds in your DNA and in your proteins. What's happening is with your proteins, very similar to DNA, right? You got amino acids, isoleucine, glutamate, and whatever. And this is folded up like this for a reason. We've got hydrogen bonds forcing these guys to interact the way that they're supposed to in a protein. You introduce too many H pluses, that protein breaks down, just like cooking an egg. Works the exact same way, denatures the protein. We're starting to track why this stuff is big picture stuff. We're starting to see how it connects. All right, so when your patient has a blood pH of 7 instead of 7.35 to 7.45, look what's happening to the proteins in their body. We start unraveling them. They're supposed to live at 7.35 to 7.45 for a reason. Yeah? Okay, good. Did they ever talk about this stuff with you guys in chemistry? I don't know why they don't put it into perspective this way, but I feel like you miss something when you don't talk about the big picture stuff. All right. Doing the math is great. What element is in all organic molecules? I know you guys know this. All organic molecules have so carbon. Now here's the question. Let's see if Cassie can answer this one. All right, Cassie, are you here today? Cassie. Okay, you're Cassie. If I were to put this particular element on here, bear with me while I draw it. You guys know this one, right? CO2 or carbon dioxide. Now, I'm going to tell you that it is not organic. It's not. Why? Kind of. So what do we need in order for something to be considered organic? We need to have another thing with it. We typically need, and it's okay, because you talked about it, so I'm going to give you credit because we're thinking about it, okay? We need to have hydrogens there too. So if we start talking about carbon dioxide, but if I started drawing this, does anybody know what this one is? I'll make an easy one. CH4. You guys think methane. There it is. This is methane. Methyl ethyl propyl butyl, right? Methyl ethyl propyl butyl. That's about as far as I go. Okay. Methyl ethyl propyl butyl. So if you have one carbon, it's methyl. If you have two carbons, it's ethyl. If you add an OH group, that makes an alcohol. Ethanol has an OH group at the end of it, etc. So it's bringing back some of that organic chemistry stuff. We're getting there. All right. So this has some hydrogens on it. So we would consider methane to be organic, but not carbon dioxide, not truly. And that's just because the properties of what we consider organic molecules to be usually have to have a hydrogen interacting with our carbons. Yes.
Sometimes you see that a carbon that's double bonded to an oxygen, and it has all four of those electrons. So that we know it's
should be, but it can be tricky, right? So let's say, let's get a different color because we're starting to get all one color on here. Let's say I draw you this. Here's a carbon. I'm gonna draw you something a little bit complicated. There's something going on out here. Who cares what? Okay. Here is some nitrogen stuff going on. And here's what you're talking about. Is that an organic molecule? Yes. That's why. But literally. No, that's what determines it. Does anybody know what this is? I'll put this here to try to help you out a little bit. Amino acid. So I don't need to put the R there. We should be seeing this. And what makes it amino? This, right? Amino is the NH2 group. You all thought you were getting away with this. Quit lying to yourselves. All right. This is what we call carboxylic acid, right? This is a CO2 that's attached to something. CO2, and it's got an H on it in this case. But that H, remember, if you learned in chemistry, can kind of switch between. And what happens is, if it gives up that H, it can bind to another NH2 over on the other side, and et cetera, blah, 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 and make another amino acid linked to it, right? If it did that, we would call that a peptide bond, right? All right, so what were you going to say?
It'll find a way to get that hydrogen.
So if it doesn't have the hydrogen, it'll be linked to something else because it wants to share an electron with somebody. Yeah. If it gives up that H, that O will go negative. It'll still have stolen that electron because those oxygens, they will have their electron. They will have their electrons. So they're going to steal it from somebody or hog it from somebody. Okay. So sometimes you'll see that drawn as C. We'll just draw it. We'll do it like this. Sometimes you'll see it drawn as this. Somewhere in the milieu, there's a last so lonely little sad H plus that got its electrons stolen from it. Okay, that's what's really happening with our carboxylic acid. It donated the H plus into the milieu, if you will. And then we're left with a couple of the O with a negative. That's an ionization event. Yeah, that's ionization. So that can be a covalent bond, but it can be a very tenuous covalent bond. And then when we do steal it, that would be ionic at that point. Yeah. Okay, we're tracking. All right, so we got our organic molecules. They got to have some hydrogen in it. We got into that general concept here. We're going to move on, and I'm going to leave all the junk up here. I'll remove it if I need to. True or false? Water is a poor solvent. Stomach is starting to hurt. Don't need that. Okay, so this is false. I'm glad we agreed on that. So I'm going to call on Evelyn. Evelyn here today? Okay, Evelyn. So can you tell me why water is a good solvent? What is it about the properties of water that makes it a good solvent? It has to do with the molecule. I'll point that out. So I'm trying to circle it, but I keep forgetting that it keeps closing my drawing thing. Here's water. So I'll help you out a little bit here. Focus on this guy here. What is it about that you think makes it good at mixing with other things? There we go. So it can attach to other molecules because it's polar, right? We got a little positive going on and we got a little negative going on. Anything else that has a little positive or a little negative to go with the thing. Or remember, opposites attract typically, right? So anything else that's polar interacts well with water, which is also polar. As it turns out, there's a lot of polarity going on in our bodies, which is why water is a solvent of life. There we go. Now we're getting there. We're thinking through it. Good job. Next question. What is water's pH? So it is up here. It might be hard to see, but it's up here. Most of us know this one. So there's one of them up here that is alkaline. There's two acids, one neutral and one alkaline. So Dylan, which one's alkaline? Exactly, right. So there's another question about pH. We'll let that do the talking next. So 12, that's fine. Next. Good job. Alkaline, same thing as base, right? What's the range on the pH scale if you're an acid? You guys are doing great. All right, so we know less than 7 is acid and greater than 7 is alkaline or base, right? So I'm not going to ask a question about this one because I kind of just did. Lipids. Tell me about lipids. There is all of the above as well, if you feel like only one of them applies. Lipids do. All right, this is all of the above one, and I'd say majority of our votes went to that one. But anyway, they obviously make up the cell membrane. We got phospholipids, right? And that's our lipid bilayer. So we draw our lipid bilayer real quick. This is another one that comes back to that polarity. And don't you love it? Because I'm not going to shut up about it. But chemistry really is our backbone of my microbiology. So you guys know what a phospholipid bilayer looks like, right? We got a couple of these charged heads. If I draw the thing, you guys recognize the thing, usually. It's got 2 tails on each of these, but I'm just going to draw one because I don't want to waste my time, okay? And we know the charged heads are on the outside and the tails are on the inside. And those tails have a property that we call, anybody remember? Hydrophobic, thank you. Hydrophobic, water-fearing, because these are all hydrocarbons. Now, I told you oxygen steals that hogs electrons and even steals them, right? If I draw you this, and I've got only H's coming up, I'm not going to draw each H, so bear with me. I'm going to represent them. Okay, these are all H's. In these bonds here with carbon, it's always equal. There is no hogging going on. Now, what that means is those type of molecules that are a whole bunch of carbons and hydrogens, and there's equal sharing, they're so at peace with each other. There's no polarity at all. That doesn't mix well or jive well with the polar parties. That's so much so that no matter what you do to mix your oil with your water, it never will mix completely, right? It'll always separate back out. That's how strong hydrophobicity will override anything else. Because carbons share equally their electrons with their hydrogens. These hydrogens, they don't have slight positive or slight negative or anything. Those hydrogens are completely, totally at peace with the carbon that they're sharing with. Total equal sharing. Not like oxygen, right? Okay. Anyways, so when we have our cell membrane, like we do down here, these heads, we just talked about this, right? So these are hydrocarbon tails, a bunch of things at peace, but they're attached to something. I know I called them a phospholipid, but what does that phospho really represent? Phosphate, right? Anybody remember what a phosphate is? How many O's? That's a lot of oxygens to hog electrons, man. In fact, PO4 is a 3- because it's hogging that many electrons. Now, if I draw you this, A, adenosine, and there's a sugar, it's usually ribose. We got an A attached to a ribose, and then 3 PO4s. What is this? You might not usually draw it this way. The A is adenine. The ribose is a sugar. And then we've got 3 phosphates. Each of them has a 3 negative. Oh, roughly. Okay. When we got the covalent bonds, it's a little different than that. But we got to keep in mind, these things are some negative suckers. You guys know what this is. ATP. Adenosine triphosphate. As it turns out, there's adenine combined with a sugar. When the adenine is with the sugar, we call it adenosine. Okay. The triphosphate is the 3 phosphates that are linked together. And if those are hella negative, it's like South Pole to South Pole magnets. They are constantly repelling each other, but they're covalently bonded to each other. They have to share some electrons with each other. So because it's oxygens associating with that, they're like, I want to hog. No, I want to hog. And so they're like constantly fighting with each other, but they have to share the electrons just to be an atom. And they hate it. And they're pushing against each other the whole time. So you cut off one of those phosphate groups, it releases a ton of energy for the cell to use. Your body relies so heavily, and I think I mentioned this last time, but we're going to bring it back to this really quick to keep the chemistry side of things into perspective. Your body runs out of ATP so quickly that if I suffocate you, you die in a couple of minutes. Even though you are thousands of cells with multiple mitochondria in each of those cells that are designed to make ATP as your energy source, you run out in minutes. That's how much your body relies on ATP. All because it cut off oxygen, which feeds into your electron transport chain in aerobic respiration. Now, I know some of you guys are thinking like, oh my God, aerobic respiration, oh, the Krebs. It's not coming right now, but it is coming in chapter 10, which is unit 2, okay? And we'll review that stuff, but you are welcome. That I don't make you guys remember that acetyl coenzyme A binds with oxalic acid and then makes the citric acid. And then that gives up an electron and it becomes the blah, 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 blah. We don't need to know all that. I'll let you know what you need to know for the Krebs cycle when we get there, okay? But it's gonna be the principles, not the different. I'm not as interested in the different chemicals. That's not as important to me. What is important is if we think back to that stuff, glycolysis makes pyruvate. That's the point of glycolysis. Y'all remember glycolysis and how many steps were in it? We went from glucose, the 6 carbon, to 2, 3 carbon pyruvates. And there's like a million, like 6, phosphoglucose, whatever BS that's in there, right? You guys remember that from chemistry? The whole point that you probably didn't even take home from that is that glycolysis makes pyruvic acid. That was the whole point of glycolysis, just to make pyruvic acid, because pyruvic acid can be turned into acetyl-CoA, and acetyl-CoA enters the Krebs cycle. The whole point of the Krebs cycle is to steal electrons from that. And the whole point of stealing the electrons in the Krebs cycle is really it. All of that, yeah, that's really it. The Krebs cycle is just to steal electrons. And when we're stealing the electrons, we'll make some carbon dioxide as a byproduct. That really is it. And we steal the electrons in the form of the NAD and FADH2. Does this sound familiar to you guys a little bit? Vaguely. We take those electrons and shuttle them through the electron transport chain. That's where we really make ATP. We make 34 ATP there. We only made 2 net in glycolysis. We made 1 in each Krebs cycle. Who cares? Because in the electron transport chain, we're making 34 suckers. Okay? So that's the goal, to get there. Then you guys are going to learn about fermentation. And fermentation only goes through glycolysis. That's it. When you ferment something, you never go through the electron transport chain. So they're only making their energy, our fermenters only make energy just in glycolysis. We only get 2 net ATP out of that. Wow, what a loss of ATP, right? But they'll stay alive, and your own muscles will do that too. If you work your muscles to the point where you're not getting enough ATP to them, they'll ferment to make a little bit extra, just to make it through that lift or make it through that run or whatever it is that you're doing when you're exercising. But that acid that we make as a product of that fermentation damages your cells and leads to pain and achiness. That's the lactic acid that gets built up. So this is all big picture stuff, but we're seeing how it relates to the big chemistry stuff. We'll come back to aerobic respiration and fermentation in chapter 10. That is unit 2 stuff. But I want you to keep all this stuff in mind. This never goes away. This isn't stuff that is separate from what we're learning, right? But it connects all to this. This is helping kind to see the big picture stuff with this. We talk about lipids here. This is where we got off track. I say me. I promise this is all relative, right? But we had our lipid bilayer. Our cell membrane is made up of phospholipids, which have a phosphate head, which is negatively charged with the tails on it. It's a lipid still, but it's got a charged end and it's got a non-charged end. So Jonathan here today? You're Jonathan. Okay, Jonathan, can you tell me what we call a molecule that has a charged end and a non-polar end? See if you can remember the word. I'll help you. It starts with an A. Think of frogs. Frogs, what type of animals are, what do we call frogs? You guys want to help them out? What amphibians? Okay, so stick with the first part of that. So when we think of something that has a charged end and a non-polar, I'll even help you even further. A hydrophilic water-loving head and a hydrophobic tail. Something that has both of those. We call them does anybody remember? Amphipathic. There we go. So phospholipids are amphipathic. So we got there. They have both going on, okay? All right, we'll come back around because we got very few people and a lot of questions. So I want to call on you guys about that stuff in a minute. Okay, cool. I'm getting heartburn. All right, it's never a good sign. All right, hormones. When we think about sterols, kind of most of us, maybe especially if you've taken physiology or maybe even anatomy, I don't know how much they teach you guys about this, that a lot of our hormones are based off of the sterols. Okay, so sterols are lipids. And one of the most common sterol is cholesterol, which I realize is not a hormone, okay? But it is very important in your membranes, in your cell membranes. When we draw a membrane like that, it's also got a bunch of stuff stuck all throughout it. Either that's channels to help things through, or things like cholesterol that give a little bit of rigidity. So eukaryotic cells have cholesterol in the membrane to give rigidity. Whereas like fungi, they have something called, well, you guys probably heard of it. It's called, oh my God, what is it called? Now I can't remember. Ergosterol, something like that. Come back to it later because I can't remember the name. And luckily I won't test you on it or else I would remember it, I promise. But anyways, it's related to cholesterol, but it's a different thing. I think it's ergosterol. It definitely starts with an E. I feel bad that I don't remember it. Anyways, So different groups might have different kinds of things supporting their membranes. Making energy for the cell. We can actually, and you might have heard about this in your biology, or not biology, your GLB classes, your organic chemistry classes, whatever you took before this, that you can actually go through something called gluconeogenesis. And that's just a fancy word for taking other things that are not sugars and turning them into some aspect to feed them into glycolysis. So usually glucose or something in those steps. And we can actually do that with fats. We can take parts of fats, especially the glycerol part of like triglycerides. We can take that, break it down and feed it into the glycolysis or Krebs cycles, just depending on where we are with that molecule. So we can use them to make energy. In fact, you can get 50 ATP out of the average fatty acid. It's wild. 50. Whereas glucose gives you 6. Now this should put into perspective why eating fats causes you to gain more weight than eating carbs. And when you eat proteins, you get less ATP out of proteins if you're feeding your body with protein. So you're filling your body up with something, but not getting a lot of ATP out of it. So you want to keep your ATP up, you can eat more proteins than you can carbs. You eat more carbs than you can fats. Because if you have a little bit of fat, you get a lot of energy out of it. In fact, so much that your body doesn't know what to do with it, so it just stores it. Right? Right. Okay. We're doing great. All right. Which of the following, look at this screen. Which of the following organic molecules is amphipathic? We just talked about this one. You guys were paying attention. So amphipathic means we have a charged head and a non-charged tail. It's usually long, okay? So the phospholipids fall into that category. Nucleotides don't. They've got a lot of charged guys on there. Amino acids don't. We got a couple of weird charged ends on it. Can we have R groups, though, in amino acids that are non-polar? Absolutely. Right? We'll talk about that in a little bit. Yeah, some of the amino acids might be amphipathic. Some of them might, but are all of them? No, not all of them. All right, what is a monomer of a nucleic acid? This means the things that make up a nucleic acid. The parts that build the nucleic acid are called. So nucleic acids, like DNA and RNA, are polymers that are made up of smaller subunits. And those subunits are our nucleotides. Okay. Let's see. Do we have Jillian here today? Jillian, can you tell me what the 4 nucleotides, you don't have to name them, but the letters that represent them would be fine for DNA. Mhm. Right. Excellent. So it's okay. You'll get there and you'll at least be able to recall them again because we'll learn about those in chapter 6. That's a unit 2 thing. So we'll revisit all of that stuff again. Do you remember what A binds to? Yes. So I remember that quite easily because C and G look like each other. I think they do. So you can think of AT and T. And that's so I think that's what I thought you were going to say. But what I think too is that A and T kind of look like each other too. They've got harsh lines in them, whereas C and G got the roundness to them. I know it's silly, but that's how I remember it too. Okay. I have a biochemistry degree, so if I'm thinking of it that way, it's okay for you to do it too. All right. Let's move on. You guys are doing so good. All right, alpha helices and beta pleated sheets are examples of which level of protein structure? I know. We've got the primary, secondary, tertiary, and quaternary. Which one has the alpha helices and beta pleated sheets? I'll talk to you a little bit about protein structure in a little bit and how I remember how it works. I'll try not to draw too much on here. And I'll remove this stuff before we move on to chapter 4. Don't worry. Okay. So this is our secondary level of protein structure. So let's see, Sonia. Okay. Do you know what the primary structure of a protein is? Always almost say it. Do you remember? It's okay if you don't. Okay, so we can come back to you. No worries, because like I said, we'll have plenty of other things to revisit. So I'll help you guys out with this, okay? Think of a protein as being spelled out. So what are proteins made out of? What are the subunits? Do you guys remember that? Amino acids. Okay. So one amino acid, another amino acid, another amino acid. It doesn't really matter what they are. That could be any amino acid. Spells out your polypeptide. okay? When you're reading your RNA, remember codons and stuff. Again, we'll revisit this stuff if you don't recall it. This chapter 6 revisits all of that, okay? You'll take 3 nucleotides, AUG, on your RNA. Remember, U replaces the T on RNA. AUG codes for methionine. You always start with methionine. Then the next 3, AAA, I don't know, codes for phenylalanine or something, okay? And you'll go on and on like that. So you'll take your methionine amino acid, your phenylalanine amino acid, and you'll join them by your peptide bonds, and they'll get linked up together and whatever. You're building your whole ribbon. And that's your polypeptide that you get, okay? That's your primary level. Now, you have a whole bunch of hydrophobic, or you have charge that are positive charge or negative charge, or polar that are positive polar or negative polar. And they're next to each other on your ribbon. Now they're going to start causing them to repel each other or want to be near each other or whatever. And so that's what leads to the alpha helices and beta sheets. Think of it as taking a literal ribbon and curling parts of it. Parts of it are curled and parts of it are straightaways. That's the alpha helices and the beta sheets. It's a sheet, straightaways. Your tertiary level is taking that ribbon that you curled parts of it and now tying it into a bow. This is governed by farther away amino acids interacting with their environment and each other from farther away. So what typically happens is the cell and your body and everything around it is in water, right? And we already talked about water is polar. So if you have a whole bunch of hydrophobic amino acids, they're going to want to hide inside and let the polar guys be on the outside. And that leads to our folding, like we saw up here, right? So we'll have hydrogen bonds between some things holding things together. We'll have polar things on the outside, non-polar things generally on the inside, and that'll lead to your overall protein shape. And we'll also have the disulfide bonds that'll create covalent bonds to hold some parts of it together. Okay, so now you've tied your bow. But that's not the end, is it? Because we still have quaternary structure, don't we? So your quaternary structure is whenever we either add your gems to the bow, which will be like cofactors, which we'll learn about in chapter 10. So don't think too hard on that, okay? Other things that help the protein function. Or sometimes when you're making a pretty package, you don't just use one bow, do you? You might pile up on a few bows to make a big bow. That's your quaternary structure, okay? You guys are doing great. All right, here's your podium for chapter 2. We're going to remove all this junk. Okay. Good time too. All right. Beautiful. So now you know Jessica's the one to beat. We will see if I can get out of this. We're going to be pulling up, hold on, you can sign out, it's not a big deal, but we are going to pull up chapter 4 while I'm thinking about it before I forget. We're not going to start it yet. We still got to finish chapter 2. So I'm just going to go ahead and pull it up. If you want to stay signed in, you can just leave it in the background. Okay. All right, so our exercise for chapter 2. It's basically just questions. This one's questions. Chapter 4, as in case you couldn't tell, is an actual real life journal article study. We'll get to it. Let's stick with our questions for now for our chemistry stuff. How are electrons distributed in a molecular bond between a highly electronegative atom like oxygen and a hydrogen atom that is covalently bonded? So this is a fancy wordy way of saying you got hydrogen bonded onto an electron. How are those electrons distributed? So Sonia, you want to try your hand at this one? So this is talking about the oxygen and what it wants to do with the electrons versus the hydrogen. Excellent. Yep. So we can see here too, when I'm forcing you guys to think about the stuff and talk about it, it makes you think about it a little bit differently, doesn't it? So you can hear me talking about it and maybe understand what I'm saying, but when I'm forcing you to talk about it, it gets you guys to work your brain a little bit differently with this stuff, right? So excellent. So I know you know that. Verbalizing it is helping a little bit. This is basically getting to the idea of the oxygen hogging the electrons. It wants to keep them. That's the idea. That really is just that. What is happening to hydrogen concentrations in a solution if the pH goes down? Haley. pH is going down. What are the hydrogens doing? Are they going, I'll say this, are they going down or up? So if the pH is going down, can you tell me, are we getting more acid or more base? More acid. Okay. So does acid have more H's or less H's? Yeah, you go. Okay. So we're getting there. So we're connecting those ideas together, right? So pH goes down, H's are actually going up. And that's because of that equation that we looked at earlier, okay? Because it's the inverse. Just remember it's the inverse. It'll kind of help you remember what's going on with that stuff, okay? Good answer. All right. All righty. The 4 groups of biological macromolecules. We'll go ahead and call on, is Julisi? Is it Julisi? Is that how you say it? Julisi. You guys have some names in this class. Sorry. Okay, so Julisi. Go ahead and tell me, if you can name all 4, that's great, but just name at least a couple of them. Excellent. So carbohydrates, lipids, the proteins, and the nucleic acids. Excellent. Next question. Give one example of a hydrophobic molecule in biology that we discussed in this chapter. Jessica.
Possible.
Easy enough, right? Phospholipids. We've also got cholesterol. We've got fatty acids, triglycerides, those sorts of things. All this one to lipids. There's actually one in the next chapter that's actually going to be pretty significant. It's a wax. Well, that's hinting at it. If you know, you know. Okay. All right. Last question for chapter 2. Given what we have learned about proteins and their structures in this chapter, why might a decrease in pH negatively impact a protein's ability to function? This is the type of question that I might ask on a short answer question on my test, right? So we've talked about this multiple times today. So then putting this into words. Does anybody want to try? I won't call on somebody for this one because it's a little wordy. Jonathan.
The extra hydrogen.
Mhm. Yes, so breaking down the proteins because it's interfering with their hydrogen bonds, right? Especially. So it's exactly right. All right, excellent, excellent, excellent. All right, we're doing great. You guys are doing awesome. Almost called on everybody, I think. There's just one. Like Duncan, I haven't called on you yet. Yeah. Yeah. So again, when we go through chapter 4's Kahoot, volunteer answers. And if I, if we passed over somebody or, you want me to call on you again, I mean, just don't let me stop you. Right? So anyways, obviously this is very conversational. It's meant to be this part. So we're going to jump into chapter 4 here in a second. If you need to take a minute, take a breath, take a breather, take a stretch, we're going to jump into chapter 4's Kahoot next. So make sure you are signed in. That I'm getting acid reflux.
Can you eat those?
I probably should have Tums. Does anybody have some?
Yep.
Yes, Tums would be perfect. Yeah. You are. I like to hear it. Thank you. Put my gum out before I do. Gum is probably not making anything better, but I had this Italian sandwich in side section of the around there. Yeah. Yeah, they are good. Mhm. So I entered in the grades for these. You don't have to really submit anything for them. Yeah. As long as you're here participating. Yeah. So if you guys ever missed a day like this, if you had a reason to miss, like let's say your kid was sick or something like that, then you get a doctor's note and email me, then I'll tell you how to make it up on your own. You'll have to do a thing for it, right? But if you're here, then just showing up and participating, you'll get full credit for it. That's actually what I'm doing over here, is putting your scores in.
So even those. That you were placed on.
Yeah, those. Yeah, you don't have to submit anything unless you weren't here, and then I'll ask you how to fill them out. Beautiful. We'll get going once we get everybody back here. Take a breather. All right, so let's go ahead and get going. Dylan's still out there, but we'll be all right. All right. First question for the Kahoot. And I'll probably ask Duncan a question about it. We'll see. True or false? Since bacteria don't have a nucleus, they can't have DNA. True or false? Okay, so this one's false. Most of us got this one. So great job, everybody. So Duncan should be pretty easy one. Can you tell me what a nucleus is? Well, how would we define a nucleus? So we could say their genome would be their genetic makeup. So we're thinking in the right realm. So when we think about eukaryotes and akaryotes, what's the difference? You know? Okay, so that's all right. So eukaryote, U means true, akaryote, A means without, and it used to be called prokaryotes, which was before. So if you could heard it either way, akaryote and prokaryote are the same thing. Okay. So prokaryotes or akaryotes, same, they lack a nucleus. They still have a genome, they still have genetic info in the form of DNA, but they don't have a nucleus. So what a nucleus is a membrane-bound barrier. It's a membrane barrier around that genome. So in bacteria, their genome, their DNA is just floating in their cytoplasm. Now it is found in a certain area of the cell. We call that area a nucleoid, but it is not bound by anything. It's not separated. Whereas our cells, you may have learned this in your chemistry classes, but our cells keeps our DNA inside of our nucleus, inside that membrane. And when you copy your DNA, that'll happen in your nucleus. When you make DNA into RNA, which is your working copy of a gene, that happens in the nucleus. But then we take the RNA and we bring it to which part of the cell? Do you guys remember what it was called? Can you abbreviate it even? The ER, the endoplasmic reticulum. Oh, you guys know this. And that's where the ribosomes read it, right? And turn it into protein, and that's translation. So that happens in the cytoplasm. We got to get the RNA out of the nucleus to do all that. And again, we got chapter 6. It's going to go. So no worries if you don't remember that stuff, but that's what happens in our nucleus. But bacteria, they don't even have a nucleus. They can literally, while they're transcribing and making their RNA, their ribosomes can start making protein before they're even done making their RNA. They can just what we call co-transcriptional translation. We'll learn about that chapter 6. So I might call on you again, Duncan, just to give you another chance to prove that you know what we're talking about. All right, the peptidoglycan layer. In gram-positive bacteria compared to gram-negative bacteria. Now, this really is our bread and butter, so I will take some time on this one. Now we're really getting into the bacteria, not just cells, but our bacteria. I really should not have eaten that sandwich. It's official now. All right, so we can recognize, most of us saw that it is thicker. So I'm going to draw you a little picture, okay? Here's a cell, and here's a cell. Both have an inner membrane. That's what the pink is going to represent, okay? Inner, what's another word for the inner membrane? Cytoplasmic membrane. Because it's holding the cytoplasm in, okay? All right. Let's get another color here. Gram-pos, let's make it purple. It's really going to be hard to see the difference. Let's just go with yellow. It's easy to see. Here's our wall. This will be our peptidoglycan wall. In gram-positives, we have a very thick wall. Gram-negatives have a tiny baby thin wall. And they've got another layer outside of it. It's the same. It's a membrane. Outer membrane. All right. Do you remember gram staining, Duncan? Or is that gonna be a lot to ask of you? It's okay if you don't.
Like from the lab? Or-
Yeah. Like either from the lab or from your stuff, because this was in chapter 4 too. Do you remember what color the gram-positive stain?
It's a
Close.
third.
Close. Do you remember what the gram-negative stain? Okay, it's okay, because we're gonna learn that stuff. We're doing that in the lab on Thursday, so you actually get actual practice with it. So it's okay if we don't really recall that just yet, all right? But I'll show you a little bit about, so peptidoglycan, so I'll ask you this general kind of question, okay? Glycan, what does that suggest to you? What kind of molecule? So we think of our 4 macromolecule types. We have lipids, carbohydrates, proteins, and nucleic acids. What does glycan hint to you? What does it feel like to you? Yes, what do you think? Sugar feels like sugar. Okay. Maybe because we think of glycogen, right? Because of sugar. What does peptido feel like to you? Okay, excellent. Protein. If we break down proteins even farther back, it just brings it back to that same word. They'll be like peptides, right? So we have little amino acids, a little stretch of amino acids. We just call that a peptide. So AA, right? Okay. What happens in peptidoglycan? I'll actually draw it over here. Bear with me because I'm going to try to be better about this. Last time I drew just blobs and it got really ugly really fast. So we're going to try to draw some hexagons here, which you guys remember about sugars looking like hexagons, right? Aren't you glad that I'm not making you do like Fisher models and all that stuff? And like I said, I have a biochemistry degree and I don't want to do that stuff. All right, so we have these strings of sugars. So these are our sugars. And we have a whole bunch of layers of this, not just 2. It'll be like layer on, layer on, layer on, layer. All right, so it's just sugars that are bonded together. And these are special sugars that I won't test you on. You're welcome, okay? But we'll put G's and M's to represent them. And what they are is N-acetylglutamate and N-acetylmuramic acid. But that's what they are. But they're sugars, and they're just alternating with each other. And these sugars, M and G over and over again, M, G, and whatever, these sugars are cross-linked across from one another with bridges. These are peptides. They're like 7 amino acids long that just hold the long strings of sugar together. So you can see pretty quickly how this side is attached to another level of sugars and this side's attached to another level of sugars. We're building a scaffolding, right? And that's peptidoglycan. So in gram-positives, we have many layers of this scaffolding. In gram-negatives, we might have just one. Very thin. We're talking 30 to 300 times thicker for gram-positives. It depends on the species, but they're quite a lot thicker, okay? All righty, so when we stain these guys, we use something called crystal violet to stain with. We use purple here. And the stain gets trapped in these pockets of our scaffolding. So that stain is crystal violet. I'll put a CV for that. In gram staining, we'll add crystal violet. It gets trapped in the scaffolding. And then we use iodine, which I'll use brown because iodine's kind of brownish, right? We'll use iodine to kind of link it into place. It fixes it in place. And iodine, definitely you can read that. That definitely says iodine. Iodine is a mordant. And I always think mordant sounds like mortar, like you do with brick mortar. It sticks the bricks together. So mordants stick the primary stain together. Okay, so in this case, if crystal violet got trapped in there, iodine makes it stick. Then we rinse with ethanol. Ethanol, because of its nature of having that OH group on there, will rinse away any crystal violet that didn't get stuck in place. Now, gram negatives, they don't have any scaffolding to trap anything. You can iodine it as much as you want, but the ethanol will still rinse it out because there's nothing really, no scaffolding holding it kind of in place. What happens is gram positives trap the crystal violet like this, all in their wall. And the gram negatives don't. So when we rinse with ethanol, this leaves these staying purple. That rinses the gram negatives clear. So we will counter stain with safranin so we can at least see them. And safranin is red. That's the principle of the Gram stain. We'll go over this again in detail again when we do the Gram stains on Thursday, but that was a chapter 4 thing as well. So I wanted to touch on it a little bit here. Okay. And this is, like I said, this is our bread and butter. Knowing that an organism is Gram positive or Gram negative gets you a pretty far way with bacteria. Okay. So Gram positives, positive purple, okay. Positive purple, negative red. To me, red is a negative color, so that works for me. Okay. It works for you too. Purple, positive. Let's move on to the next question. I'll leave the crap up again. Okay, you're welcome. We're doing all right. Gram negative outer membrane. So I told you it has an outer membrane. It has something special in that outer membrane. I've got a bulky old thing over here. So I don't want to get up anymore from my stomach hurts. Okay. All right, so the answer is endotoxin. I know we were kind of guessing at this one, okay? So does anybody, there is a little special molecule out here, and you might have learned it in your studies about it being called something else. Does anybody else remember what it was called in the LPS? Okay. Yes, LPS. So it's this little molecule on the outer membrane called LPS. LPS is fancy, is an abbreviation for a fancy word. Lipopolysaccharide. Again, if we break down the word, lipo means we have a fat component. Polysaccharide means we have a many sugar part. We have fats and sugars. As it turns out, the lipo part specifically, lipid A, and I won't test you on this. It's enough for me that you know that LPS is the same thing as endotoxin. And like it sounds like, endotoxin inside endo, because it's part of the gram positive, the gram negative cells, okay, gram negative membrane. It's part of them, it's in their cells, okay? Toxin, because it's toxic to us, it's toxic to our tissues. So let me tell you something about the immune system really quick. Our innate immune system, which is our neutrophils. You guys may have learned about this in physiology or anatomy if you've taken either of those courses, that neutrophils are the most numerous of your white blood cells by far. You should about 55% of your white blood cells should be neutrophils. They're very generic, basic cells that eat up bad things in the body, waste and any bad guys. Neutrophils can tell me that this is a marker. Neutrophils will tell me that that's marker. And they'll say markers are not self, eat them and get rid of them, okay? Endotoxin, or LPS, same thing, but in the medical community, it's more relevant as endotoxin because it's toxic. Endotoxin is something that helps your neutrophils say, that's a marker. That's not self. Eat it. So with endotoxin, it can say, that's a bacteria. Bad. Eat it. So we have to train your body too, because have you got bacteria in your gut? Yeah. So we can't be overreacting to those bacteria. So we have to train our body when and where it's appropriate to react to endotoxin. And typically endotoxin, when it's part of an actual cell, our body doesn't react to it. But when it's broken up, then your body will react to it. And they shouldn't be broken up in your gut. They should be living happily in your gut, right? So it's another way that it'll know. But if it sees endotoxin hanging out on itself, excuse me, marker, what are you doing? You're not supposed to be here. And your neutrophils will eat it. Your adaptive, acquired, whatever you want to call it, immune system, your B and your T cells. Your B and your T cells don't recognize that it's a marker. Your B and your T cells say, that is the red marker with the black thing stuck to the back of it. That is the green, the blue expo marker. That is the green expo marker. Your B and your T cells know exactly what it is. And your T cells are the captains that govern everything. And your B cells make antibodies to the green expo marker, to the red marker with the black thing on the back. It doesn't have a label. Sorry. All right. So those guys, your B cells can tell the difference between specific pathogens. So specific, in fact, that to your B cells and your T cells, the flu that you got this season looks like a completely different thing to your B and T cells than the flu you had last season. And that's why we need new flu shots every year, because they look so different to your body. there's part of your body that might be able to recognize it's the flu, but it's not strong enough to fight back like your B and your T cells are. And your B and your T cells remember. Your neutrophils don't. They're just walking around saying, oh, that's a marker, eat it. But they don't remember. But your B and your T cells do. Your bombers and your tanks is how I call them. Okay. We'll talk more about those in Unit 3 because your immune system is wildly awesome. It is so cool and so undertaught. It is really cool. Okay. All right. And I feel like you're learning from somebody that really learned their immune system. And so hopefully you guys can benefit from that. But even when you stub your toe and it hurts and it gets red and it gets swollen, you get a bruise and we have clotting and everything like that, all of that's part of your immune system response too. Inflammation in general, not just illness, like infection. Moving on. Let's get more bacteria stuff in. What is acid-fast staining? Comes back to what we were talking about earlier. Let's see if we can figure it out. Getting up again. I almost thought I was going to rest and not get so excited. You guys see how excited I get about this. Just wait till we get to the immune system. Running over my coat over here. All righty. Acid-fast staining is staining to determine whether or not mycolic acid is present. I mentioned this earlier, but I didn't name it. I said we're going to talk about a wax that is found on some bacteria in chapter 4, and this is it. Mycolic acid is a wax. So it's a lipid. Waxes are lipids. And so they are hydrophobic. And so we have to use special stains. The stain I'll use is called carbofusion, and we'll do it in lab 4 next week. Okay, but basically looking for that wax. And certain kinds of bacteria have this wax. Is either Brooklyn or CeeLo ever show up? No, neither of them? Okay. All right, so we'll just be hoping that you guys are going to like ask questions and participate is what I'm saying, okay? So acid-fast staining. So whenever there's 2 genera that can have mycolic acid. One of them I will never test you on, and that is Nocardia species. And really almost never medically relevant. Okay, so I just don't talk about them a lot. So they won't be on your test, FYI. The other one is Mycobacterium. We'll write 2 things on there that people often confuse. Mycobacterium. Are you real? I'm tapping on my iPad. I'm losing it, guys. I guess I am that old after all. You know, we were talking about, somebody was saying to my other lab earlier today, they're talking about how they feel old, like when somebody didn't remember like 9/11 or something. I don't know. Let me tell you, I remember Chernobyl. How about that? How does that make you feel now? It should make you feel young. Does anybody know when Chernobyl was?
Girl, we're like the same age.
I barely remember Chernobyl, but I remember a little of it. So if you actually remember Chernobyl, you've got me beat. But I have a memory of it. I remember it being on the TV and my parents being worried about nuclear fallout because they were worried that it would blow with the weather. Chernobyl happened in 1986. I remember. I know I don't look like it, but okay. So mycoplasma, I did type it all. It's just this is janky keyboard. Mycoplasma and Mycobacterium. All right. When we see Mycobacterium, I want you to think about the second part, okay? Bacterium. Does that say anything special about it? It doesn't. Then you'll go to myco. Now the myco tells you that it has mycolic acid, which is that wax that we're talking about there, okay? I'll make this clearer. If you see mycoplasma, we'll start with the second part, plasma. Now I want you to think about this. Imagine this is a cup that is open, okay? I'm not going to do it with an open cup because it would make a mess everywhere. But imagine this is an open cup and I'm walking around with somebody's plasma. We can agree that plasma is the liquid part of the blood. It's mostly water, you know? All right, so I'm walking around with a cup of plasma and I drop it. If it was a cup that was open, would that plasma maintain its shape of the cup? It would spill everywhere, right? Same thing with our mycoplasma. Think about the plasma part. Plasma means doesn't hold its shape. Why don't these organisms hold their shape? Does anybody know why they don't? Why mycoplasmas don't hold their shape? Anybody know? Yes. They don't have a cell wall. Trucking, you're doing it. Okay. Doesn't hold a shape because no cell wall, none at all. So mycoplasmas are gram negatives. They don't even have that little yellow. They're just two membranes. So they don't hold their shape. They're what we call pleomorphic. Looks like this. Means pleo is many, morph is shape. They don't have a defined shape. They're not cocci. They're not bacilli. They're pleomorphic. I don't know. They have blob things going on. I don't know. Right. Rando shapes at any given time. Both of these are medically relevant. All right. Can anybody name one disease caused by a mycobacterium? Tuberculosis. Anybody know another one? Ooh, we're getting somewhere. So not quite. We'll come back to it. Okay. Yes. We'll come back to that. Right. Okay. We're thinking about a thing. So leprosy. Mycobacterium will be in the name of the organism that causes these diseases. So the bacteria, Mycobacterium tuberculosis, speak for itself, causes tuberculosis. Mycobacterium leprae causes leprosy. Okay. Let me give you an example of a mycoplasma. It is called Mycoplasma pneumoniae. And of course, if we're typing it and we are just perfect and we do everything correctly, we would italicize, but it didn't let me do that. Sorry. It just deleted the thing. So pretend like that didn't happen. We'll just write the thing. Mycoplasma pneumoniae. Now, if we gave a shot at this, we could probably guess at what disease it causes, right? Pneumonia, but it's more specific than that. So the most common cause of typical pneumonia is Streptococcus pneumoniae, which is also the most common cause of bacterial meningitis. It gets around. Okay. But Mycoplasma pneumoniae, still relevant medically. It still does cause pneumonia, but it causes a more familiar one, actually. We often think of pneumonia as being something that will knock you down on your butt and you're in a hospital bed. Typical pneumonia presents that way. But we've had plenty of people that we've known that have, what do we call it when they're around and about and they have pneumonia? Walking pneumonia. That's Mycoplasma pneumoniae. You were familiar with it, you just never knew. Okay. Good. So if it has mycobacterium are the only ones you actually are going to get tested on that are acid fast positive. They're acid fast because they got that wax, and that stain looks for that wax. It's a wax found on gram positives. Some mycobacteriums are gram positives, or one of the gram positives. Okay, moving on. We're going to keep going. If you guys have any questions and need me to clarify something, just let me know. No, you're fine. What was the question? Oh, 650387 if you get kicked out. Next question. Mycoplasma, it is up here, is an acid-fast positive organism with a thick, waxy cell wall. Is that what I said about mycoplasma? I think we called on everybody today, yes? Did we get everybody? Okay. All righty, so this one's false because the mycoplasmas, they don't have any cell wall. They are not acid fast. The mycobacterium are that. So I know they sound very similar, but they are different. And of course, you're going to have to know the difference for the test. Right. All right, next one, question 6. What are bacilli? A few cell shapes we had to know. I'll give you a little hint there. It's a shape of a cell, bacteria cells. What's bacilli? All right, so this is not my pen. I don't need to be putting that in my mouth. They are rod shaped. Okay, they are rod shaped. Anybody know the name of round or spherical? Cockeye. Okay, so that's the actual shapes, and we'll review that in the lab today. We're going to be doing microscopes today. Our next question, what does peritrichous mean? We have flagella are the only appendages the bacteria use for motility. If you're motile and you're a bacteria, you got at least one flagellum. Okay. Flagella is the plural. There's different arrangements around the cell. Many bacteria aren't motile. When we talk about Staphylococcus aureus, it is non-motile. Some bacteria are motile, and when they are, they have a flagella. If you have one flagella, we call that monotrichous. Trich means hair. They're hair-like appendages. And they move in a rotating fashion. They don't whip. They can't control them like this. They control them just like this. Okay. They rotate either clockwise or counterclockwise. So if you're going along and you're trucking along as a bacteria, you're spinning them in one way. When you shift the spin the other way, that's the tumble. You guys remember about runs and tumbles when you guys were reviewing this stuff? All right, so that's the tumble. You have a whole lot of flagella all around you. You might look something like this. Here's the cell. Oh no. Here's the cell. You have a ton of flagella coming off like hair blowing in the wind. All right. You can control each of them individually. They can. Pretty wild. And which way they're rotating or going back. And that allows them to steer a lot better the more you have all around you. When you have them all around you like this is peritrichous. Monotrichous looks like that. This is not a very good example, but okay, we'll just actually do lophotrichous. Lophotrichous is a tuft. Lopho. No. Peri. And amphi, I want to draw a really good rod here, has both, one at each end. Amphitrichous. Up. All right. Bacteria don't have any other way to move. They don't have cilia. Now, we're used to think of amoebas. You guys remember about amoebas? One thing we generally pick up usually from like high school or grade school or biology or something is that amoebas move like this. And then they drag themselves along. And then they drag themselves along with their little blobs that they stick out. We call them pseudopods. They're fake feet, pseudo-fake podia, like podiatrists' feet. Okay? Bacteria are not amoebas. Amoebas are eukaryotes. So eukaryotes can move by pseudopods. Eukaryotes also have flagella, but eukaryotes can whip them. A lot more directed movement there, right? And eukaryotes can have cilia too. In fact, your gut cells have cilia, but we can actually use that for feeding, but also some of them use it for movement too. And so you could have 3 different ways you can move as eukaryotes. But if you're a bacteria, if you move, you have at least one flagella, and that's all you could. That's it. Fimbriae are for attaching to surfaces. Pili create bridges between the cells, and they can shuttle genetic information between them that way, which we'll learn more about in chapters 6 and 8. All right, any questions about those appendages? Moving on. Just doing so good. All right, bacterial capsules. Tell me about them. Which of these apply to the capsule? This is not the wall. This is not the envelope. This is something else entirely. Let's see if I can write with the pen. We'll do the pen. Okay. All right. So we got this majority went with the right answer. So capsules are a type of glycocalyx. Calyx just means cover. And what does glyco insinuate again? Sugar. Okay, sugar carbs. Sugar covering, quite literally. Okay. And so cells often have some sort of sugar covering, a glycocalyx. In fact, eukaryotic cells do as well, but we call that our extracellular matrix. It's what keeps our tissues individual tissues. So our extracellular matrix has a glycocalyx, and it also might have collagen and hyaluronic acid and whatever else is in your extracellular matrix. But these guys, these often have a sugary coating. And it's quite impressive. When we look at the image here that it showed us, now look at the size of the cell compared to that white area around them. And we're talking like twice the thickness of the cells. Now imagine you had 2 people thick of honey around you. Now you go in and rob a bank, how are them cops gonna catch you? They're gonna be grabbing onto that honey and they'll never be able to get to you because that honey is in the way. If it was that thick of honey around you, 2 people's worth of honey. This helps you stick to things. Of course it does. That's what we learned in the chapter 2. That are glycocalyx, whether it is tightly bound, like a capsule, or very loose, like the slime layers. So that glycocalyx helps with sticking to surfaces and with building biofilms. So biofilms are, for example, like the plaque on your teeth. Okay, to remind you about it, because now you're going to want to brush your teeth. But we know that's a biofilm, right? We know it's bacteria that kind of build up on layers and feed on the gunk on your teeth. And then they make acid and that decays your teeth. And that's how we got to dental caries being the most common of the infectious diseases that we know, right? Well, that's a biofilm. And these bacteria are pretty smart in their biofilms. They can actually communicate with each other. What they'll do is say, oh, it smells like somebody pooped over there. They'll get chemical signals like you would smell, like we talked about previously. I don't know if we did actually, it was a different class, I think maybe chemotaxis and moving towards things. When we talk about your flagella and how you move towards a signal or away from a signal, depending on what that signal is. So if you smell a plate of cookies up here, that's a chemical signal that you're getting with your brain that I don't want that. I'm gonna go get me a cookie, right? But if I had a pile of dog poop up here, I'm gonna move away from that. Thank you very much, right? Those are chemical signals that you are moving, and that's chemotaxis. You're moving in response to chemical signals. Your bacteria will do the same thing. If they're smelling, it smells like somebody died over there. I don't think this is a good place. I think I'm gonna go somewhere else. That's how they communicate in the biofilms. They pick up on chemical signals like that. And that's what we call quorum sensing. So it's just sensing how the quorum, the group, is doing. And it really is that simple, picking up on the chemical signals. It's not as complex as us talking to each other. We're just reading the room. Yeah. All right. Anyways, so I didn't want to leave you guys out of that because I talked about it in other classes. So we want to talk about that. When we're talking about the capsules, though, building that biofilm layer in which you'll be communicating like that is helped by having that sticky coating. You can stick to the other bacteria and help others stick to you. But the third thing that does, adhering to things and creating biofilms, the third thing that it does is protect against phagocytes. Anybody know what a phagocyte is? Yes. A white blood cell. It's going to be white blood cells. Your white blood cells eat up the bad guys, like we talked about with your neutrophils. Neutrophils are a phagocyte. They eat up things. Phago means eat, and cyto means cell. Okay, so eating cells. Phagocytes include your neutrophils. Eosinophils and basophils can do it too. Mast cells can do it a little too. Not as interested in them. Definitely your neutrophils. They're 55% of your white blood cells. And one other biggie are your monocytes and their derivatives. This is going to include your macrophages. I'm not going to test you on that stuff for Unit 1, but we will definitely get into it in Unit 3. And if you've ever heard of monocytes or macrophages, you'll see why they're so important because we can't do anything without these guys. Okay. All right, so moving on, this protects against that being eaten up. Oh, and let me tell you, almost all bacteria have a capsule. There are plenty that don't, but I'd say the large majority of them have one. So our immune system has to be having something in place to deal with that. And it does. Don't worry, it's got you. Our next question. When are endospores produced? Another one that people often miss. Uh-oh. Question yourself. Question your choice too. Endospores. When are we actually making the endospores? Go away. All right. So endospores are made in nutrient. I'm so glad you guys got this one. Or you took the message and you're like, okay, it's probably not warm, moist conditions, because that's what everybody else was picking. So I'm not kidding. So we went with nutrient deficient conditions, and this is absolutely true. So endospores are a hardy, dormant, inert structure, and it is not a method of reproduction. So what happens is we go from normal cell and we switch into endospore, which is like a really hunkered down version. And then we switch to normal cell. The normal cell is called vegetative. Vegetative cells are active they're multiplying. These are the ones that make the toxin. These are the ones that are growing to make your infection and everything like this. The endospore is really just waiting. It's completely dormant and completely inert. It has to be turned back into the vegetative form in order to do anything. If you're going from vegetative version into endospore version, this is a process called sporulation, making the spore. If you're going from endospore to vegetative, this is a process called germination. You guys understand what germination is because we understand about plants germinating. When you germinate a seed, it's growing, right? So if we're going from endospore to vegetative, we must be going to the growing form, which is the vegetative form. Okay. All right. The endospores don't do anything other than wait. And one cell becomes an endospore, and endospore becomes one cell. So it really is just there to wait things out. And when we talk about disinfection as opposed to sterilization, you guys remember how we discussed the difference between the two, that sterilization is killing everything. And disinfecting is killing pretty much most things, but some things don't get killed. The thing we usually leave behind, endospores, because they're too hard to kill. And in order to kill them, we need some really harsh things. And so when we're disinfecting, it usually gets rid of everything but the endospores, usually. Okay. So this is why containing certain hospital infections can be such a problem, because some of them are associated with endospores. So there's two genera. So if we talked about Mycobacterium and Mycoplasma, or the Mycobacterium is acid fast, it has mycolic acid. Mycoplasmas are the ones that don't hold their shape because they don't have a cell wall. Some other ones that we want to be aware of that are kind of separate. These are endospore formers. Bacillus species, SPP means species, okay? So it'll be in the name, Bacillus blah, 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 or it might be Lactobacillus blah, blah, blah. If it has bacillus in its name, number one, you know what shape it is. That gave it away. You know it's a rod shape. But also, number two, if it's a bacillus species, it makes endospores. Another one, and we called it earlier. Do you remember what it was? We talked about the disease. Do you guys remember the disease that was associated with it? Called it out. Okay, well, I'll tell you, I'll help you. Clostridium species. Clostridium species include C. tetani, C. botulinum. Sounding pretty bad so far, right? C. perfringens. That causes, you're going to kick yourself when I say it, gas gangrene. Right? And then the other one, another one that I know, if you guys work in a hospital, you know this and you hate it. C. difficile, or C. diff. All right. The disease that C. diff causes, we're so used to calling it just C. diff in hospitals, but the disease it causes is actually called pseudomembranous colitis. If you hear itis in anything, it means inflammation is being caused. That's what itis means. So colitis is inflammation of the colon. And there's a pseudomembrane that is formed, quite literally like a membrane that's made in the colon by these bacteria creating a biofilm. Now here's the thing. Bacillus species, the main most medically relevant bacillus species that I could bring up, we're going to talk about in just a minute, is B. anthracis. Anthracis causes obviously anthrax. Don't poo-poo on anthrax, okay? Anthrax is very serious. The most tame version of anthrax you can get is cutaneous anthrax. So it's a sore on your skin. And it causes something called a black eschar. It's eschar, but I just say it eschar because that's how I remember how to spell it. Eschar, okay? And it literally is black. It's black like the lid of this pen is black. It's a black like scab. I don't mean it's dark purple. I mean it is black. And it's because of the necrosis that the bacteria vegetative cells are causing. But the endospore gets in the wound. It says, this is warm and moist. This is pretty good. I got some nutrients. And then it comes back to life. It only takes them to go from endospore to vegetative cell 1.5 hours. Right? Impressive, right? So hunkered down and completely inert, to get into a dividing cell within 1.5 hours. You step on that nail and you get that Clostridium tetanus inside of your body. Now, the key is with this, the Bacillus species, you see I only have one listed here because that's the only one that's definitely medically relevant. We'll talk about some other ones later on, but they're not as complex significant as this, okay? But these are all aerobes. And like they sound, they need air to live. We'll talk more about that in chapter 9. But Clostridium species are all anaerobes. They hate air. They will die in air, okay? All of them. But the endospores won't. The endospores are there to survive the air. So you get C. diff. It's hard to clean it. It's resistant to any of our cleaners. You can't get rid of it. It's not killed by any sanitizers or anything like that. And the endospore itself is also resistant to most antibiotics. So you take antibiotics and it clears out all of your stadium seats in your gut. And suddenly C. diff multiplies up. You might have only had 1 or 2 C. diff guys taking up a seat here or across the stadium. But all these seats are cleared out and they're waking up and they're like, huh. They're coming out of their little endospore going, and they multiply out and they take up all the stadium seats. And now you've got pseudomembranous colitis. And now that person's pooping out endospores and vegetative cells. They hit the air, vegetative cells die or turn into endospores. And the endospores are what is being transferred around the hospital. All right. And then any patient that you have that's on antibiotics will get.
Because these are Clostridium, we could be like in lab, we would do the slant ones with them because they're.
So, right. So when we're working with Clostridium species in real life in labs, we can't work with them in oxygen at all. They won't grow at all. Not in our incubators at all. They'll just stay endospore and they can't divide and multiply like that, right? So we can't culture them. So if we want to grow Clostridium species, we have to have a special vacuum container that removes all of the air. It's crazy, but yeah, so we can't study them in our lab. Yeah. So you guys remember, if you worked in hospitals, anybody ever seen anybody do any blood cultures? They're those bottles that they have, right? So we usually do 2 of them, right? You usually have an aerobic one and an anaerobic one. This is why we do the anaerobic one. It's for stuff like this. Make sense? Cool? Who's doing good? Tracking. Yes, you wash your hands with soap and water to mechanically remove the C. diff, right? That's the idea. So you don't spread it from patient to patient. And then you just got to hope that you got them all off of you. And it's hard. And in patients with C. diff, and if you've had, like my mom has C. diff, okay? She has diverticulitis. It's very common to get C. diff when you have diverticulitis. And so what we do is we treat it with something called metronidazole. Metronidazole is used for anaerobic conditions. Now, metronidazole doesn't work in the presence of oxygen. And also really mostly affects bacteria that are anaerobic. So for other bacteria might be okay, might be. And it should attack the things that are anaerobic. And the idea is that if we kill enough of the C. diff, we can repopulate with enough good guys. Can we ever completely eliminate the C. diff with just some antibiotics? Probably not, because those endospores. And in fact, removing C. diff entirely is very, very difficult. And it might even take radiation chemotherapy to remove it. And then we can do that. But then what we have to do is repopulate your colon with some good bacteria before C. diff comes back in. And so the way we do that is with fecal transplants. You've heard of them, but they're actually much more common than you realize. And yes, we literally take somebody's poop from somebody that you live with typically, because it's more likely to be successful that way. And we don't make you eat it, okay? We put you under anesthesia and through a colonoscopy, we'll deliver it into your colon. Okay? We're getting somewhere, putting things into perspective. All right, last question of the Kahoot quiz. How would you describe the strepto arrangement? Strepto. There's a couple of arrangements we talked about. Staphylo, strepto, and palisades are probably the biggest ones. Pairs is diplo. I'll give that one to you. Pairs is diplo. What's strepto? The way I remember it, strepto sounds like strip. Staphylo don't sound like strip. So I don't get it confused. But staphylo is our grape-like clusters. Strepto is chains. Pairs is diplo. Then we have palisades, which is fence posts next to each other. Not like this, but like this. Strepto, palisades. All right. This is our podium, and then we're going to do really quick our little activity. Then we'll go to lab. Ah, this time Jillian's the one to beat, guys. So I'll let you guys know. My online class for the lab, we'll do the lab quiz later. I'll talk about it later. Let's do this activity really quick. It's not BS anymore. Let's clear up our screen too. All right. Investigation of inhalation anthrax case in the United States. Anthrax is a naturally occurring disease. I won't read the whole thing, I promise. We're going to hit the major points, okay? Naturally occurring disease caused by bacterial spores. We more specifically call them endospores, okay? But bacterial spores in the soil. And as it turns out, anthrax is a zoonotic disease. Anybody know what that word means? Zoonotic. Animals. Yeah, literally. It's found naturally in animals, okay? So this is caused by the bacterium Bacillus anthracis, which we brought up just a moment ago. So we know immediately Bacillus, because it's Bacillus, we know that it is endospore forming, which it's talking about that in the case. So that's not a giveaway or anything. Endospore forming. It's called Bacillus, so we know what shape it is. What does Bacillus mean? Rod shaped. There's another giveaway that I didn't talk about in the Kahoot thing, but that we can pick up on from this stuff too. If you are Clostridium or Bacillus, these are the only guys that can make endospores. These are the only ones. They are both gram positive. So if you are gram negative, we know automatically you can't make endospores. But do all gram positives make endospores? So Staphylococcus, Streptococcus, those are gram positives, but they don't make endospores. Only Clostridium and Bacillus species do. But one thing you can know for sure, if it's a Bacillus species, if I'm talking to you about Clostridium perfringens causes gas gangrene, you hear Clostridium, your brain needs to be thinking endospores, gram positive. Immediately, okay? And what's gram positive mean? Can anybody tell me real quick? What do we know about something if it's gram positive? Thick wall, stains purple in the gram stain, okay? Great, okay. All right, so it's a zoonosis, which means that it's found naturally in animals. Now, this person was on a road trip, a three-week trip in North Dakota, Montana, Wyoming, and South Dakota. Heavy farm animal area, so lots of farms. Animal anthrax is sporadic or enzootic. Enzootic means naturally found in animal populations and not as commonly in humans. But these people, they drove through herds of bison and burros, okay, frequently stopping with animals around the vehicle. And then the man got what we call pneumonic anthrax. Anthrax, or another way you could write it is pulmonary. Anthrax. Okay, so we talked about anthrax a little bit earlier. We talked about the cutaneous version, right? And that's the best one you can get. 20 to 30% mortality rate in the cutaneous. So when we're talking about pulmonary anthrax, it just gets worse the deeper in the tissues that we end up getting. It's harder to treat it. So we're talking probably a 50 to 70% mortality rate with pulmonary. Okay, so this is bad. And I don't remember if the man died or not. I don't know. So I didn't read the whole thing. I'm just being completely honest here. I just got the information that we needed for this. Okay. But anyway, so this is investigation into that. And I don't have the whole thing, but if you want to go look at it, please don't let me stop you from going and reading the whole case study. Okay. But anyway, so this man has this. Now these lines are here that are going to be completely useless because we're going down to our next question part. Okay. Questions around this. We have the info that we need. We know it's anthrax. We know it's a zoonotic disease, and we know it's got endospores. So let's answer some questions about it. What is the disease caused in the case study? So this is pulmonary anthrax, right? And what is the scientific name of the pathogen that causes that disease? That was Bacillus anthracis. If we're handwriting it, you underline it. Remember? Okay. It's pulmonary anthrax. Pathogen that causes the disease, because I didn't want to give it away, okay? B. anthracis that we were discussing here. Is it gram positive or gram negative? It doesn't say in the case study, but is it gram positive or gram negative? It's gram positive. We know this, gram positive, because it makes endospores. And because it's bacillus, and I told you that, endospores, and they're gram positive, and so we can say that, okay? Because of that. That's really the only way we know that. How would you describe that envelope? Gram positives have what kind of outside to them? Thick cell wall. Do gram positives have an outer membrane? Nope, no outer membrane. Do they have endotoxin? No. Excellent. All right. Does the pathogen discussed in the case study have a nucleus? Does Bacillus anthracis have a nucleus? Because it's a bacteria. No bacteria do. Okay. And what's its genome made out of? DNA. So this one's a little tricky for some people. So we did talk about that a little bit. Bacteria do have DNA. It's just not in a membrane-bound nucleus. But it is still just double-stranded DNA. All right, so we're gonna clear this up just a little bit. So I gotta scroll down. The pathogen discussed in the case study has endospores. What's an endospore? Does anybody want to try that one?
Dormant.
Yep.
Born.
It's just a dormant, hardy version that waits out the conditions to be woken up again. So it's dormant. It is completely inert. So I want to stress that because the next question asks, when the pathogen causes the disease, when this person actually gets pulmonary anthrax and they're having disease, and the toxin is being produced that leads to that development of the disease, is it the endospores that are making that toxin? No. Okay, so we're tracking with that. Some of us, I see us going, no, no, no. Why? How do we know it's not the endospores doing that? Because the endospores are literally inert. They cannot do anything. They can't make toxins. Making something needs energy. In order to get the energy, you have to make the energy. That's doing stuff. They ain't doing anything. The second they start doing, they become vegetative. The vegetative version of the cells are causing the illness, making the toxin, etc. Are we tracking? Okay, that's it. So we're done with lecture for today. Excellent work today, guys. We're going to head up to the lab. Whenever you guys get up there, you can take a little break if you want to. But once everybody is up there, we'll go ahead and get started with the lab. Good job. I don't need to see my face